effects of the supposed miracle weight drug, or also known as DMP, and has been recorded to have fatal effects on people, and they take different doses.
So DMP is a drug that people are using to lose weight. However, based on our research, the drug is destroying the fat cells and the muscle cells. So in the cellular respiration chemical equation, at the end, you're making energy. But when you insert this drug into your body, you can't make any more of that energy. So that's why you can see in this graph, you're still consuming O2, but then you stop making ATP, which you need. And then also because of this drug, heat is being released into your body, which is harmful. And then this is why one of the lady passed away, and this is because the lady was losing a lot of weight, but then her body temperature had risen to above 100 degrees. So she was taken to the hospital, and this is what had happened to her. So her body stopped producing ATP.
What organelle in the cell is targeted? Anybody knows that?
Mitochondria.
Okay.
Mitochondria, because the cellular respiration and everything we've been going, mitochondria is the powerhouse of the cell. It's the powerhouse because it makes the ATP. So that's why the mitochondria is the organelle that's being affected.
Right. So do only muscle cells or fat cells have mitochondria?
No.
Every cell. So if you're taking DMP, are you only targeting fat cells?
No.
This is an overview of the compound, DMP for short, so pronounce this it's a yellow crystalline structure. It's sweet. It's easily dissolvable in organic compounds. It's composed of like a benzene ring with a hydroxyl group at the 1 spot, and at the 2 and 4 spot it has nitro groups connected to it. So I'm going over oxidative phosphorylation, and as you can see I have AB, and they're just balanced chemical reactions that kind of very simplistically explain what's going on during the oxidative phosphorylation step. As she was just talking about, the NADH does get reused starting right here at the beginning of this step during the electron transport chain, involving hydrogens and oxygens, and I'll go over that in a little bit more detail, followed by a phosphorylation step in which in the end result is your ATP. So, can I have 4 volunteers? Let's get them from the back first. So in this first part, the NADH comes out of the Krebs, like Adi was saying, and that's the first part of this electron transport chain. So go ahead and be the first complex. So she has our electron. She's our first complex, and all these complexes they're just proteins, a bunch of proteins in a system working together to get a job done. And the first electron will get transferred to her, and then she will transfer it onto the next complex. So now we're at complex 4, the electron is going to start doing some stuff here. So first of all, the electron right there will give it to an oxygen, and I'll go ahead and be that oxygen. So now I'm oxygen, and I'll have the final say in this electron. And at the end of it, I'm going to grab 2 hydrogens. So now I've created the water. But I still haven't created the ATP. This is only the electron transport chain. This is only the first step out of the 2 steps that I talked about. As you can see, the hydrogens were getting shot up out of the complexes into the intermembrane space. They originally came from the mitochondrial matrix at the bottom. So when the electrons were passing by, each time it moved from one place to another, it shot a hydrogen into the intermembrane space. So now we have a whole bunch of hydrogens passing up there. But just like osmosis, eventually it'll get too concentrated of hydrogens up there, and they'll want to come back into the mitochondrial matrix to balance themselves out. Just like everything in chemistry biology, we have to have an equilibrium. Eventually the pressure or the concentration will build up in there, and it'll need to come back. Unfortunately, it can't just diffuse through the intermitochondrial membrane because it has an impermeable layer. Basically, it just can't go through it. So luckily, we have ATP synthase, and let's just make him ATP synthase now. The hydrogens can pass through the ATP synthase, and that's good. So those hydrogens will come back through him, and the ATP synthase also has a toll feed. So it's saying, I'll let you back into the mitochondrial matrix, but I'm going to use some of your energy to bind ADP and the inorganic phosphate, PI, to create ATP. So when the hydrogens start coming back through the ATP synthase, ATP is created, and the hydrogens are back in the mitochondrial matrix, and it'll just keep going in a complete equilibrium and just keep passing by and coming back forward.
actually giving these beetles DMP?
No. What we did in the experiment is we mimicked the effects of DMP, like what happens with the body, like in humans. We mimicked, like they heat up, right? So we saw how it affected the metabolic rate in the beetles. And so how we did that was we used this metabolic rate apparatus. Here we have the Vernier LabQuest. Here we have the Vernier CO2 sensor. This is the Nalgene biochamber, the 251, and the best beetle. And then this is also the Go temperature Vernier temperature sensor. And that's how this experiment kind of works. This is like our first trial, and this was just at room temperature. This is just to see how the beetles' metabolic rate was at room temperature. And then later on the next day we did, we put this biochamber in in a water bath, and then it was a heated water bath. So we saw how it compared to when they were they were hot, so to say, and how that affected their metabolic rate.
The first one was at the room temperature, which was 24 Celsius, and the second one here is when we put it in the heated water.
Wait, can I add something real quick? If you notice on there, when she got the linear regression, she did find the R squared. So the standard deviation, when they're closer to 0.99, that means the more accurate that your results were, which she got, our experiments, our results were 0.97 and 0.96. So they were about as accurate as we expected them to be. For the CO2 that was being released in room temperature and when it was heated.
Let's just quickly sum up our findings. So just by looking at the amount of CO2 produced, we kind of indicated that the metabolic rate did increase due to temperature increase, so it did support our hypothesis. Going back to what Juan said, if we were to get a chance to redo this, we would have more trials for the O2 consumption in the first, just like with regular temperature, with room temperature. And then again, with heat applied, we would just have more trials so we can get better numbers. So I kind of just put there heat applied increase, CO2 also increased, thus metabolic rate increasing. The heat applied was mimicking what DMP does, so that's why the CO2, the CO2 release support our hypothesis because of the temperature increasing.
Our final recommendation to the FDA is to ban this drug, prescription, or over-the-counter for humans. And beetles.
And beetles.